So thank you for the introduction, and it is a privilege to be here today. And I will be sharing the research on defining the transcriptome of male germline stem cells. So since this is a reproductive biology crowd, I'm going to keep my introduction very brief. So spermatogenesis is a classic model of stem cell biology, adult stem cell biology. And spermatogenesis stem cell generates millions of sperm every day. Spermatogenesis stem cells reside in the spermatogonia population, which is right here. And any disruption to the spermatogenesis stem cell population in the function, reduce in function or loss in function, results in infertility or subfertility in male. So infertility, approximately 10% of all reproductive age couples worldwide experience infertility, and half of those cases attribute to male factor. So 1 in 10 of those men experience azoospermia, where the ejaculate is completely empty of sperm. So 90% of those cases are idiopathic, where there's no known cause. So by understanding the establishments and movements of the spermatogenesis stem cell pool, we can establish different techniques to treat and diagnose male infertility or subfertility. So just a basic schematic, the spermatogonia population encompasses both the undifferentiated population and the differentiating population. Within the undifferentiated population resides the asingle spermatogonia, which encompasses the spermatogonial stem cell. Spermatogonial stem cell then replenishes the progenitor population, which includes the non-stem cell, asingle, the A-pair, and A-aligned population. And again, spermatogonial stem cell sustains spermatogenesis, and the progenitor cells will then differentiate go into differentiation and become the differentiated spermatogonia. So further clarification on what a stem cell is. Stem cell is defined by the ability to regenerate and sustain a developmental cell lineage. And for spermatogonia stem cell, it is defined by the capacity to regenerate and sustain spermatogenesis. And the transient amplifying progenitors, which includes the non-stem cell, asingle, A-pair, and A-line, they do not have regenerative capacity, so they actually contribute to a steady state condition. So currently there is no marker for the spermatogonia stem cell population, so it really hinders the ability to study this population. So first, my lab went about to try to define a spermatogonia stem cell marker, so it facilitates us in studying this population. From previous study of the Oakley lab, we have found that the inhibitor of DNA binding for ID4 is a putative marker for spermatogonia stem cell. It is only expressed in a selective population of asingle. As you can see here, PLZF, which stands for the spermatogonia, which includes asingle, A-aligned, and A-pair, is in green. And then ID4 is in red. You can't quite see them, just follow the arrows. And so here, the arrow head shows the merged image of the subpopulation of spermatogonia that is both same with ID4 and PLZF, whereas the big arrows are PLZF only. So it shows that ID4 is in the subpopulation of the spermatogonia population. So to further investigate ID4 as a putative marker of spermatogonia stem cell, we generated a transgenic model called ID4GFP, which we inserted the eGFP coding sequence in frame with the exon 1 of the ID4GFP transgene. This mouse is then maintained in a black 6 background. we designated it as LT11. And the expression of ID4GFP is found in both testes, brain, and kidney. So here is cross-section IHC staining, and it shows that by using anti-GFP antibody, the ID4GFP expression is enriched in 6 days after birth, and it slowly declines through 12 days after birth to being very rare in adulthood. With live imaging, here is actually stained with Lin28 antibody. Lin28 stains for pneumonia, so you can see the A single, A pair, and A aligned cells. And here is a live imaging of the ID4GFP cells, and you can see that they are only staining for the A single population, confirming that ID4GFP is a cumulative marker for pneumonia stem cells. So for further quantification and quantitative characterization, we did cross-section, and within those cross-section, we stained it with ID4GFP antibody. It shows that in 3 days post-birth, you can see that 86% of the cross-section tubules has ID4GFP staining, averaging at 2.5 cells per positive tubule. And there's a slow decline as the mouse age and develop into adulthood, when adults with only 2.4 cross-section tubule has the ID4GFP positive cell, and averaging at only 1 positive cell per positive tubule, indicating that the stem cell population is enriched in neonatal and slowly declines and become very rare in adulthood. So with this cumulative marker, we then wanted to find out what is the regenerative capacity for ID4GFP cells, because stem cells are defined by the regenerative capacity. So next, we crossed the ID4GFP LT11 mice with a ROSA mouse, so that all germline express the LAC-Z transgene. So in the presence of XL, it will be stained blue for visualization. This ID4GFP LAC-Z mouse is then, the testes are extracted and we made a culture with island selection, so we're enriching for the undifferentiated spermatozoa, which is especially useful to have a primary culture of undifferentiated spermatozoa because the limited amount of spermatozoa stem cells and also progenitor that you can extract from one testes. By doing this, we can increase the number for analysis. So within this population, approximately 10% of those cells are ID4GFP positive. As you can see here, this is just bright field, and you can see, well, there is the little dot, and the word is right there. So next, we took those culture cells, and we used fast sorting to separate out the GFP positive population and the GFP negative population. We then respectively put the ID4GFP population in one testes of the recipient mice, and then the negative population into the other testes of the recipient mice. And then after several months, 3 months, we extracted the testes, treated with XL for visualization. Remember that each blue colony is a stem cell that regenerated into a colony of spermatogenesis. And you can see that with the ID4GFP population is enriched in stem cell capacity, whereas the ID4GFP negative population is depleted in stem cell capacity, indicating that the ID4GFP is the spermatogenesis stem cell population, whereas the ID4GFP negative population is the progenitor population of the spermatogenesis. So now that we have this method of separating out stem cells from the progenitor, we wanted to study the transcriptome between the 2 different populations and defining the stem cells, spermatogenesis stem cells transcriptome. So these 2 fast sorted populations, ID4GFP and ID4GFP negative, are then used in the Germline platform for RNA sequencing. The RNA contents for both populations are revealed. This information is then analyzed with the top hat, which is actually the tuxedo package would include top hats and cufflinks, which you will align the reads, into the this genome of the mouse. And then those mapped reads are then using our statistics and also cufflinks. You will generate differential expression of genes. And with the differential expression gene analysis from tuxedo and RNA-seq, we found that only out of all those genes, 10,000 of them does not show differential expression. And then the ID for GFP negative population, the progenitor population, has 25 genes that are differentially expressed. And then within the ID for GFP positive population, the spermatogonia stem cell population, there are 12 genes that are differentially expressed. And within those 12 genes, they include genes that encode proteins that regulate high RNA metabolism, cell growth and metabolism, intracellular signaling, and transcription. So in conclusion, this body of work shows that ID4GFP is a marker for spermatogonia stem cell, and that spermatogonia stem cell state is regulated by a subset of genes, so that there is a difference in transcriptome between progenitor and spermatogonia stem cell population, and spermatogonia stem cell state is regulated by a very small 12 gene group. So for future analysis, I will start looking at the biological relevance of these 12 genes and how they go into establishing the spermatogonia stem cell pool, and also how they maintenance the spermatogonia stem cell state. So with this, I would like to thank the OT lab, especially Dr. Oakley, Melissa Oakley, and the Peter. Thank you. Yes. There is very few of them, and when I looked at the different sets, especially there are the metabolism sets, they actually are very different. So I don't know why there's so low of them, but it's pretty useful to have so low to investigate, so I don't cast a line there. And the p-value that I cut off is quite small.
So I'm not asking the number of cells.
Oh, why is it so?
It's not what I expected.
You're expecting more stem cells. Well, each stem cell will go into progenitor, which is 2, and then they go into like 2 to whatever end that is amplifying. So I think as long as it's not damaged, there's no reason to have too many stem cells within the population that is in the steady state. Was that it? Yeah, Charles.
I was curious if you compare the gene expression from your spermatogonia stem cell population to stem cell populations in other tissues, and seeing if there's any similarities or differences.
Not that, but that's a good idea. I'll probably do that soon. Catherine.
So why do you think there are so many ID4 positive cells in a neonatal testis versus an adult testis? So what are you thinking of looking at what distinguishes those cells in the neonatal testis versus the adult ID4 positive population?
So your question is why is there such an abundance and frequency of ID4 within? I think it's because they're still establishing the steady state, and those cells need to, as they go into adulthood, those cells will transition into the progenitor population. So they all go into pretty much an enrichment of stem cell, and then as they proliferate, then they can decrease in number once they reach a steady state. That's why I think it is. Yes?
What do you 12 mean?
Thank you.
Other questions? If not, let's thank you again.